Welcome to Trial Site News Podcast Series. My name is Adrian, and today we have with us Dr. Alejandro Soto-Gutierrez, MD, PhD, here to talk with us about an intriguing concept that the UPMC McGowan Institute for Regenerative Medicine is spearheading. They are growing genetically modified miniature human livers. Now, Trial Site News picked up this story, and I must say we are absolutely fascinated with this concept of genetically modified miniature human livers as a method to emulate human liver disease progression and support ongoing therapeutics testing. So, Dr. Soto-Gutierrez, welcome.
Hey, Adrian, thank you so much for having me. It's exciting that you give me the opportunity to talk about my projects. And I guess you can call me Alex. It's easier.
That sounds great. So Alex, can you tell us how you first got involved in genetically engineering human cells into foundation 3D liver tissue that mimics NAFLD?
Sure. It's a very long process, right, to figure out that you want to genetically engineer human livers from stem cells. But, the long story short, I have a lot of interest on finding ways to treat terminal liver failure, especially patients with cirrhosis that have NASH or alcoholic cirrhosis and failure. And, there are very few opportunities for these kind of patients that need liver transplant. So I've been looking for models. I've been looking to understand the disease for many years. And I found out that there are not really many human models that we can actually use. And access to human biopsies is also pretty difficult. So what I decided was to make my own models that have human cells where we can modify genes and study the biology of it, study pharmacology, therapeutics, and that's how we came up with this.
Oh, wow. Now, how difficult is it to actually create a miniature artificial human liver in terms of effort, time, resources, and cost?
Yeah, sure. I think 5 years ago, we thought it was really difficult, and we also thought that probably it wouldn't work. But now I can tell you that it's not that difficult. I think it's easy, and I think it's fast as well. And I think, we did all the development of deriving human mini livers from basically skin fibroblasts from people or patients. But I believe the cost is not that high compared to the benefit that we can get out of this. So in terms of timing, from the time we take a biopsy from individual and derive fibroblasts to the time that we have human mini liver, probably fatty, ready to be tested for new pharmacology, new agents, or toxicology, or whatever you want to test, it probably takes in a lab setup about 4 to 6 months, depending on the sample. So there you go.
Gotcha. And just on a side note question, what was the catalyst for deciding to go this way, specifically going for a artificial human liver? Like, how did that come about?
Sure. As I told you, mice are not humans. And a lot of especially the metabolism has to do with what the environment is around us. So basically epigenetics. And those epigenetics, those genes that are upregulated or downregulated depending on your exposure to food, toxins, drugs, etc., are very difficult to study in animals. Mice eat the same thing every day, and we just don't. So that's the way. That's how our thought process was. We need to come up with a human model that we can easily derive, that has to be personalized, because we have to incorporate as well the not only the epigenetics, which is the microenvironment, but we want to incorporate genetics into it. Whatever make us humans and make us different. So that has to be also there.
Now, although quite complicated, and so just to summarize for our audience, we will try to summarize our understanding of the potential use case reported on. So we know that you were able to seed the genetically engineered human liver cells into rat livers stripped of their own cells, where they blossomed into functional 3D mini livers with blood vessels and other structural features of a normal organ. Now, the team could then flip the genetic switch to suppress a targeted gene, for example, And this seems to be an extremely powerful way to explore solving genetic-based diseases. Could you elaborate more on this?
Sure. I think the value of this technique is the ability of controlling gene expression in these human livers. So I believe that by incorporating these inducible switches of any desired gene in human cells reflects better the metabolism, reflects better drug exposure and toxicology. So I think it's a very good biological tool to screen drugs pre-clinical trials in patients. For example, there are really few drugs that are under screening right now in clinical trials to treat NAFLD or fatty liver. But one of them is producing really exciting results, which is an FXR agonist. The thing is that from some of these drugs, only a percentage of patients with fatty liver actually respond to it. And other patients respond to other drugs, and we don't know why, right? Maybe 20%, 30% respond to one of them and the rest don't. And once you enroll in a clinical trial, you have to keep going for the next 4 or 5 years, depending on the design. So I think coming up with, making a human liver from your own skin and testing it before for all these drugs that are in trials right now, and giving information to patients to which trial will actually work better for you, it's of great value. So that's one of the main goals of this approach.
Well, and that's huge. That's amazing what you guys are doing.
And then the second goal for making these human mini livers is actually biology. As I told you, there are genetic modifications that we are basically born with. They are known as polymorphisms. And they're really small single nucleotide mutations that we have. And some of them have been related to actually development of liver failure or fatty liver. And so basically we are born with a mutation that will facilitate myself developing a certain disease, right?
Yeah.
And we have identified some of them. There are publications with large studies associating these mutations. There are other mutations that have been associated as well with protection of liver failure or development of fatty liver. So, but it's very difficult to really come up with mechanisms of what the effect of these single nucleotide mutations are. So I think the second use of these mini livers is that come up with identification of individuals that have those mutations, and then studying them in the lab, by making mini livers from their skin, having that mutation background, and then looking into the metabolism of in those cells and identifying what is the role of that mutation on metabolizing drugs or fatty acids, etc., etc. So this could lead to prevention medicine, where you can design a diet, you can design drugs before patients develop liver failure. So I think that's the second use of it.
Well, that's amazing. And there's also other applications to this down the road. I mean, you could be really changing medicine here.
I hope so. At least diagnostics and better design of clinical trials.
Yeah.
I hope that we can affect that.
Well, it's a noble goal, and I'm excited to see where you guys go with this. Now, in our summary of your study, we noted that you stated this actually was the first time a genetically engineered human mini liver with disease-using stem cells was created in the lab. Was this truly a first, and can you elaborate?
Sure. It was not reported in literature before, the engineering or generation of mini human liver with vasculature flow through it, incorporation of many kinds of liver cells into it, and basically personalized liver, where we derive, using stem cells derived from specific patients or people. So there are a bunch of technical novelties here, from the differentiation process to the incorporation and establishment of easy techniques to genetically engineer human-induced pluripotent stem cells, for example. The success rate of incorporating genetic modifications into cells like iPS cells was pretty low in between 3 to 4%. But now we found that we can very easily do it with a range of 30 or to 40% success. So it's pretty good.
That's a huge jump.
Yeah, it's a huge jump. So I think each part of the process has some novelty. So it took about 5 years of just trying, success, error, and accommodating everything. So yeah, I think it when you combine everything, it's a first. But I'm working now with other laboratories so that they can replicate this, at least the approach, and they can study better their genes of interest and the consequences. So yeah, I think that's what can I tell you about.
Gotcha. Well, and it's amazing in 5 years. Great work. Now, we did talk a little bit about some of the long-term, possibilities or achievements that you could gain through this work that you're doing. But what are some of the short, intermediate, and long-term goals you'd like to see achieved with this breakthrough? Short-term meaning 3 to 9 months, medium 9 to 18 months, and long-term anytime after 18 months.
Yeah, so the short-term goal is that we are working on right now is the incorporation of several genetic backgrounds, like I told you before. We identified already human samples from a specific patient population, and we have derived already human induced pluripotent stem cells from them, and they have a specific genetics background, a specific mutations, like I told you, a specific polymorphisms. And the short term is we're going to be making livers from those and see what the effect of those polymorphisms are. Then I think the midterm or medium term goals for this is we already started preparations to start taking samples from patients here at UPMC and the fatty liver clinic, patients that are diagnosed with fatty liver or NASH, patients that are under a clinical trial or not, and we will start preparing personalized livers from their skin and testing drugs that have been successful and that have been also non-successful in trials, and see if we can validate these mini livers to be preclinical testing to better design trials in the future. So that's our medium term thing. And the long term. So the long term is to be able to use these liver as a genetic functional screen machine, and being able to discover genes and modifications of genes that could be useful to, for example, create human livers with superpowers. So what I mean with that is that lately we've been creating human mini livers with gene editing systems inside. So that in a way that these human mini livers are editable. So then what we do is we make mini libraries of guide RNAs with specific gene modifications that we want to study, like induction of regeneration or modifications of the metabolism of fatty acids or drugs metabolism. And then we screen those genes in the whole organ, and then we test the function of the gene modification. So we are, for example, studying, like I told you, superpowers out there. We found that squirrels have superpowers. They can hibernate, and we can't. And we've been studying the genes that are responsible, especially in the liver as metabolic machine, for hibernation. And we found that, the whole system shuts down 98%. pH changes, the cells become really, really fat without getting sick. And then through hibernation, these cells are able to use that fat that was accumulated. And then 3, 4, 5 months later, they come back. And they come back with a lean liver, right? So we study the gene differences by genome-wide studies. We know where those are. We've been screening those in the human genome. And what we are using is these human livers derived from stem cells to somehow use the squirrel genes there and see if we can affect the functions of these livers. I'm not sure yet what the outcome will be. It's a fun, risky project, but it's very useful.
Well, how fascinating,
it's a
too.
genetic screen, yeah.
I mean, you look at the potential applications. Let's say, you were going to send people, out to Mars, for example, somewhere down the road, and they were able to hibernate. I mean, what kind of an amazing, all sorts of possibilities with this kind of work that you're doing.
All sorts of possibilities. Maybe we can come out with genes that are silent in our livers, and because we lost them through evolution, and then we can basically wake them up and make a liver more metabolically active whenever you need it to be. So, the possibilities are endless. So I think we have a good tool to screen all that right now.
Absolutely. Fantastic. Now, we also know, as a side note, that you started a new venture as well with Von Baer Wolf Inc., or Incorporated, and congratulations. Can you share more about how this new venture will leverage the work you did in the university to commercialize for real research use cases?
Okay, yeah, sure. It's just something new for me, and it's just extra work from the work we already have at the university. But it's also very exciting. I'm learning a lot on the sales side, like how to sell your ideas, what kind of experiments people want to see, making big budgets and stuff like that. To just bring from laboratory setup your projects into a more systematic way, right? Mm-hmm. Anyway, yes, Von Baer Wolf is dedicated to treat terminal liver failure, and we have 2 IPs and 2 projects dedicated to that. The first one, and the most important one, in my opinion, is we call it reprogramming therapy for terminal liver failure. Mm. So what we found was that cirrhotic livers that develop terminal liver failure have a different genetic identity than livers that are healthy. And we found those differences in genes. And in 2012, I'm sure people know that the Nobel Prize went to a scientist called, last name is Yamanaka, for reprogramming cells from being a doll into basically stem cells. And by forcing the expression of 4 master transcription factors. Mm-hmm. So we thought that the same principle will apply to disease, and we've been screening transcription factors that we found different between the livers with terminal failure and the normal ones, and we found a gene that actually makes a difference. And we've been treating now human liver cells coming from explanted livers of people that had liver failure, and we've been reprogramming them from being diseased into health. So that's what we call now reprogramming therapy. I think these could substitute the need to do transplants and basically treat liver failure from inside of the cells versus changing the whole organ. So anyway, that's one of the concepts. And the other concept is manufacturing of human liver cells in bioreactors. And these bioreactors are basically rats that can grow human livers inside. We are trying to grow a specific iPS-derived liver cells into these animals to produce tons of them and do pharmacological studies on them. Just, making sure that the cells we produce are fully mature and compatible to a human hepatocyte in order to make, human mini livers or whatever we want to do with them. So those are the 2 concepts.
That is amazing. Now, what do you see as the biggest challenges for the company to take off, and what are the ways to mitigate the inevitable risks faced?
I think that the biggest challenge so far is to understand how early investors want to invest on it. So normally you find people that say they are early investors and with early ideas, but whenever you talk to them, basically you have to be ready to go into the clinics tomorrow. So it's not that early, right? So finding the right investors and people with the background and the interest on the project is being difficult. So we we knocked many doors, but it's looking good. So, I have a lot of hope project, and I think it's exciting.
Awesome. Well, congratulations again. That's fantastic news. Now, anything else you'd like us to, or anything else you would like to update to the Trial Site News audience on?
Yeah, basically just want to thank you and thank your audience to have the time to listen to this. If you guys want to know more details about it, I guess you will have a link about this recent publication on how to generate human mini livers from specific patients or individuals. I'll be happy to, if people contact me or anything, I'll happy to answer questions or help them to make livers or whatever they may need.
Awesome. Yeah, I'll absolutely have links in the description to forward them directly to you. And so on that note, thank you so much for taking time out of your busy schedule to talk with us today, Alex. It's been a pleasure talking to you.
Thank you, Adrian, for giving me the opportunity.
Yeah, absolutely. Anytime. And now, for those of you in the audience interested in learning more, you can visit the company website at www.vonbearwolf.com. And we'll provide the publication website for cell metabolism in the descriptions below, as well as links for the lab as well. And then you can also find Trial Site News on Instagram, Twitter, Facebook, and YouTube. And the links will be in the description below as well. So thanks again for joining us today, Alex. It's been a pleasure.
Thank you.